I'm Zeta.
I'm Fernando.
I'm Roman.
Me llamo Tori.
I'm Magali.
I'm Cynthia.
And in this video, we'll be walking you through the physiological, chemical, and environmental aspects of West Nile virus.
Since it is a virus, the flavivirus will invade your body as any other virus. The flavivirus will invade a host cell by attaching fibers of its protein coat to a specific receptor site on the wall of the host cell. Then it will inject its genetic material, either ribonucleic acid or deoxyribonucleic acid, into the host cell. The virus then uses the host cell's machinery to make copies of the viral nucleic acid. Once multiple viruses are now formed within the bacterial walls of the host cell, it will get released, and now the body has multiple viruses invading multiple host cells.
People that have West Nile virus will tend to have symptoms usually where they were bitten. Most of the time, 75%, people with WNV don't even realize they have it, as they don't even experience symptoms. However, the ones that do have symptoms, 25%, will only experience an unpleasant flu-like illness marked by fever and chills, headaches, fatigue, and pain in muscle and joints. Symptoms often last 3 to 6 days, but it can last for as long as 2 weeks. In other cases, the West Nile virus can be very deadly, attacking the body's CNS or central nervous system. The central nervous system is composed of the brain and the spinal cord. In very serious cases, which is in 1 in 150, the virus will attack the CNS by causing inflammation of the brain and meninges or the protective membranes surrounding the spinal cord. These conditions are encephalitis and meningitis, respectively.
In meningitis, which is what most people suffer from if they are severely attacked by the virus, there is an inflammation of the meninges, which is composed of the dura mater, arachnoid matter, and pia mater, which protect the brain. When the meninges become inflamed, it causes pressure on the brain and spinal cord. The brain is enclosed by the protection of the skull, so in order to relieve pressure from the brain and spinal cord, a piece of skull must be removed. In a lot of these cases, it leads to death. Those who do not die from severe cases of the disease are commonly left with permanent muscular damage, such as a loss of muscle coordination. This is why the West Nile virus can be considered very deadly in some cases.
Flavivirus integrates with the body's cells via one of two processes, namely either apoptotic mimicry or calthrin-mediated endocytosis, the first of which includes the WNV cell absorbing a molecule called phosphatidylserine from the target cell's membrane and coating its own exterior with it, which disguises the viral cell as an apoptotic body that the target cell will then absorb into the cell. The second of these methods involves the viral cell bonding protein E of its protein coat to the host receptors and initiating a process by which the adapter and calthrin proteins bond to the viral cell and allow it to enter into the target cell safely, at which point it will release its viral DNA.
Flavivirus infiltrates the target cell's endosome, compartments used as a transport network by the Golgi apparatus, via the fusion pore model of Lee K. The viral cell bonds two fusion proteins to host receptors on the endosome, penetrates, splits, and then fuses its own cellular membrane with that of the endosome, which allows it to transport its viral RNA genome into the endosome. Here, the viral RNA is modified to be a polyprotein, a massive single protein composed of several smaller proteins, and then released back into the cytoplasm.
After this, the polyprotein viral genome is cleaved into its 13 respective components by the 3 enzymes of signal peptidase, Golgi protease, and NS3 protease, all found in the cytoplasm, and the individual components are transported to the cytoplasmic viral factories on the surface of the endoplasmic reticulum. The viral genome is then modified to be a more complex form called a dsRNA genome from its previous form, a ssRNA plus genome. This new genome is then transcribed and replicated, which then provides enough material for several copies of the original ssRNA plus genome after it is cleaved.
At this point, the virus is reassembled at the endoplasmic reticulum and buds into the cytoplasm via the host ESCRT processes. The completed viral genomes are then transported to the Golgi apparatus, where the PRM protein in the viral genome is cleaved, thereby fully maturing a fusion-competent viral genome. At such point, the viral genome is released into the cytoplasm and exits the target cell via exocytosis.
The West Nile virus originated from the West Nile district of Uganda in 1937 and was later found in Africa, Asia, the Middle East, and Europe. The virus is now found all across the USA, Canada, and Central America.
Various breeds of mosquitoes are the main form of transmission for West Nile virus.
The presence of large number of avian hosts of either viremic or amplifying nature. The presence of large numbers of mosquito species to feed on the blood of infected birds. And the presence of large numbers of other host organisms, such as livestock and humans. And lastly, the presence of antibiotics in the affected environment all affect the transmission of West Nile virus.
There is no treatment exclusively for the West Nile virus, though there are various over-the-counter drugs and antiviral drugs available to combat WNV. None of these drugs are direct treatments for WNV. There is a vaccine for horses to prevent the West Nile virus, and the virus is very lethal to them. But so far, there is no vaccine available for humans. Prevention measures against WNV include preventing the development of mosquito populations in affected regions, attempting to avert the development of stagnant pools of water in marshes, tires, and other such locations is shown to be effective in reducing the exposure to infected mosquitoes. Aside from this basic measurements of prevention of infection are to avoid going outside without long sleeves and such during the spring and autumn, when the mosquitoes are more active. The use of mosquito repellent is also effective at repulsing mosquito bites.
Because WNV is commonly mistaken for the common flu, and some people who are infected with the virus live in areas without accessible medical care, the prevalence of WNV on a world scale is statistically unknown. The disease has still been identified on every continent in the world excepting Antarctica. The virus has a total of 9 different strands which are genetically differentiable from one another and are typically regionalized to certain landmasses. However, on average, 2,765 cases have been reported per year in the U.S., from 1999 to 2013, between which years the virus experienced 2 mass epidemics in 2002 to 2003 and in 2012. About half of these cases have been neuroinvasive, meaning that traces of the virus were detected in areas surrounding the central nervous system of several patients. In the same period, there have been 96 deaths per year due to severe neurological complications of the West Nile virus. The disease is not an alarmingly pandemic proportion, but it has consistently remained a public health concern in the United States and the world at large.
The presence of vegetation in an affected region, the age of infected persons, income as related to the ability to access medical care, race distribution, prevalence of certain mosquito or bird species in affected regions, age of housing, and certain geological factors all impact the presence of West Nile virus.
In conclusion, the West Nile virus is a non-curable viral disease that originated in the 20th century and has since spread all across the globe. Though the disease is rarely fatal to infected persons, the pervasiveness of the disease to the CNS, as well as some of its intricate means of transmission, have raised concern as to the possible long-term effects of the disease. Today, treatment of West Nile virus centers on basic antiviral drug treatment, while prevention of the disease's spread is centered on preventing the buildup of mosquito populations that act as the disease's primary vectors. West Nile virus is most often asymptomatic, but but does produce flu-like symptoms in 1/4 of infected persons while causing severe neurological complications in less than 1% of infected persons. The disease makes use of apoptotic mimicry and calthrin-mediated endocytosis to infiltrate the cells of its host, and then uses the endosomes, endoplasmic reticulum, and Golgi apparatus of a host cell to reproduce its viral genome. Despite the puzzling complexity of this virus, research on the subject continues today in hopes of finding a way to neutralize this now global health threat.